Hello students, let us look at questions and answers based on our introduction to the section on nucleic acids. So let's look at question 1. Question 1 states, name the parts labeled A to E for the diagram of the nucleus that follows. So if you look at the diagram, you can see a simplified diagram of a nucleus and you have labels A, B, C, D, and E. If we look at our next question, question 2, it says name the parts labeled X, Y, and Z for the diagram of the chromosome that follows. So you can see a diagram of one chromosome and you've got the labels X, Y, and Z. Question 3 is a question of definitions. And don't underestimate these questions. They're not simply about recall. Being able to define a particular term or a concept actually shows understanding. So quite often if you are learning a more detailed aspect, like a process, it becomes much harder to understand that process, to know its logical sequence if you don't understand the basic terms. So remember, these terms are key, not just for identifying, but to understand concepts. So our definitions, 3.1, you need to define the chromatin network. 3.2, you define a chromosome. And 3.3, to define a gene. Those are your questions, right? So let's look at your answers for this very simple exercise. Question 1, provide labels for the nucleus. Label 1, you will notice, is pointing at the outermost layer. So that's your nuclear membrane. Label 2 is indicating the gaps in that outer layer, the gaps in the nuclear membrane. Those are the nuclear pores. Label 3 indicates the inside of the nucleus, what is enclosed within that nuclear membrane, and that is the nucleoplasm. Label 4 points at that dark, dense, round body. What is that? That's your nucleolus. And I just want to point out, students often make a mistake with the spelling of nucleolus. Please practice spelling, because I've often noticed that if you leave out a letter, the word changes into nucleus, which would be completely the wrong label. Okay? So take note of the spelling of the word nucleolus. And label 5 is pointing at those thread-like structures that are there. And what is that? That is the chromatin network. Right, I hope you found those labels easy, students. Let's look at number 2. Right, again, we have a diagram, a different diagram. This is the diagram of a chromosome. You would notice it's double-stranded. Do you recall at which point in the cycle of a cell you would find a chromosome visible like this? During cell division, right? So when a cell is dividing, chromosomes of this appearance would be visible. Right. So let's look at our labels. Label 1 is indicating one of the strands in this double-stranded chromosome. That is a chromatid. Label 2 is indicating the little round structure that's connecting the one chromatid to the other. That's your centromere. And label 3, you will notice, is actually a bracket. And it's indicating the entire structure. So that label would simply be chromosome. Right? Simple. But it comes up again, and you may encounter diagrams of this kind in our next section as well, where we look at cell division or meiosis. Question 3 is definitions. Right? So let's look at the first one. What is the chromatin network? We've just looked at it in the diagram in question 1. So a chromatin network is a tangled mass of threads of DNA, right? In fact, looking at the diagram gives you the definition. It is a tangled mass of threads, and you need to indicate of DNA. So this tangled mass is made up of the DNA molecule. 3.2, what is a chromosome? A chromosome is a strand of DNA which contains genes. Now, if you're asked for a definition of chromosome, you don't need to define the word gene as well. So you could simply say a chromosome is a strand of DNA that consists of segments that are genes. Question 3.3 is, what is a gene? A gene is a portion of a chromosome that provides a code for a particular characteristic or a particular protein. Or you could state it like this: a gene is a small portion of DNA that provides a code for a particular characteristic or a particular protein. If we look at question 4, the question is, name the 2 types of nucleic acids. Question 5, name 2 cell organelles that contain DNA. Question 6, during which phase of the cell cycle is DNA arranged, A, as a chromatin network, and B, as chromosomes? Question 7, state a function of ribosomes in a cell. Let's look at question 4 again, right? The question was, name the 2 types of nucleic acids, DNA and RNA. Question 5, name 2 cell organelles that contain DNA, the nucleus and the mitochondrion. And remember, we refer to the DNA in these organelles as nuclear DNA and mitochondrial DNA, respectively. Question 6, during which phase of the cell cycle is DNA arranged in different ways? If you are not sure what we mean by cell cycle, we did encounter this term in grade 10 when we looked at cell division. And basically what we are looking at is the period when the cell is dividing compared to the period when the cell is not dividing and performing various other activities. Do you recall what name is given to the part of the cell cycle where the cell is not dividing? It's the interphase. But let's get back to the question. During which phase of the cell cycle is DNA arranged as a chromatin network? Right? We will recall the chromatin network is that tangled mass. So this is during the non-dividing phase. So most diagrams of a cell that you will encounter are showing you a non-dividing phase. The next question was, during which phase of the cell cycle is DNA arranged as chromosomes? This would be when the cell is dividing. And in grade 10, you encountered mitosis, and in grade 12, you will be looking at meiosis. So those are the dividing phases during a cell cycle. Question 7 stated or asked you to state a function of a ribosome in a cell. You will recall that when we were looking at the lesson, we stated that ribosomes are not even visible on the diagram at that magnification because they're so small. But we did indicate that they are really important in performing a certain function. That function is protein synthesis. So the function of a ribosome is that it is the site, it is the actual place where proteins are built up and proteins are formed. So those were our 7 questions, learners. Let's just look at a few guidelines that would apply not only to lesson 1, but to the subsequent lessons that you would be doing as well. Now, in this exercise, you are required to label diagrams. My advice to you, in any section where you encounter a diagram, practice drawing and labeling that diagram. That's the best way to understand a structure, or to understand what parts of a structure are, to be able to identify them as well. Right? So please draw, practice your drawing. If you do practical assessments, you would be required to do drawings as well. Another piece of advice is to keep a little book. That's like a diary, like your little black book, and write down every new term that you encounter. Or it could even be older terms you've come across before, but you know we would use them again throughout our studies. And what you should do is make these habits a daily practice. So whenever you have a lesson, please try and recall what was done at that lesson. And a very good way would be to write down. When I say make notes, I don't mean make copious notes. We've got textbooks, etc. We don't need that. What you need to do is recall and write down key points that you would have come across. I will see you at the next lesson, so keep studying. Hello students, welcome to our lesson on a brief history of the discovery of the structure of the DNA molecule. Discovering the structure of DNA was like an arms race among scientists. Finding the structure was considered the holy grail in biology. Every scientist wanted to be the first to get there. You may be familiar with this famous photograph. This shows two scientists who worked at Cambridge. They are James Watson and Francis Crick, and they are analyzing the DNA model. In fact, the model we use to understand the structure of DNA and therefore its function is called the Watson and Crick model. But they were not the only scientists that were involved in the discovery of the structure. Two very significant individuals were also involved in discovering the structure of DNA, were Rosalind Franklin and Maurice Wilkins. They worked at King's College London, a rival university. In 1952, Rosalind Franklin, together with a PhD student, began producing images of DNA, and she used a technique called X-ray diffraction. And this X-ray diffraction produced some astounding images. The most famous image that she, together with her student, produced is called Photo 51. In fact, some people say this is the most important photograph ever taken. What it revealed about DNA is the 3D structure of the molecule.